All right, let's get it started. This is week number 5. We are discussing dimensionality reduction. Okay. Sounds really weird. So today we are going to cover 3 commonly used dimensionality reduction technique: PCA, TSNE, and FATE. So PCA has been around maybe 100 years old. TSNE maybe 10 years or 15, somehow like that. And FATE is a relatively new technique, maybe 5 years ago. So that's in that order. So we will have a lot of mathematical formulas throughout the lecture. This gives you the background information. So when you need to go into details, you have somewhere to look at. There are links, but this is for biologists. So in general, I want you to remember some of these facts, and there are some slides with a star on it. So you try to memorize or know, for example, how to calculate PCA, how to do this step by step. And if you want to know from step 2 to step 3, why should I do this? There are slides in there to go to the formula. There are links for the forums. People discuss why in each step in there. And the big picture is we are dealing with single cell data as so-called high dimensional data, okay? Because if you consider each cell as a sample, we are going to describe the properties of each individual cell using the information of gene expression. So there are many, many genes expressed in individual cells. So this is a high dimensional problem. So in this case, we have this table. For example, there are 4,044 genes. That means each of these cells is described using over 4,000 variables. So you can place this cell in a 6,000 dimension space. Okay, I hope throughout the previous week's lectures you already know high dimensional space, and you have some idea about where to put this, depends on the values of the gene. This concept is important. Otherwise, there's no meaning if you don't understand the high dimensionality. Why are you going to reduce that dimension? So let's put this dimensionality step in the whole pipeline of single-cell RNA-seq data analysis. Once you have this expression matrix, we normally will do this dimensionality reduction to reduce the variables that are used to describe cells. There are some easy ones. For example, if these 2 genes express almost linearly correlated, you probably can use just 1 gene or average of these 2 to replace 2 genes. So you reduce dimension by 1. Okay. Some technique we use to find the redundant information among or between these genes, and to extract or transform or summarize those covariance and replace the gene expression with something else. Okay, when I say something else, once you do this transformation, there are not any genes expression. It could be several genes expression compressed into one vector. This vector is still a variable, but it's not represent any individual gene anymore. It's a comprehensive, summarized effects. For example, we can summarize all 6,000 genes expression pattern into using 50 vectors. So you compress 6,000 into 50 vectors to represent the major component among the explaining variables patterns, okay? So this step is in between this. This will be using that to facilitate the downstream analysis. For example, you after do the dimensionality reduction, and this become a table with a 50 PC versus how many cells, depends on how many cells you have. So this matrix is smaller than before. Now you can easily to compute or more accurately compute the distance between cells. Okay, you compressed your dimension and make it small and more informative, rather than just rely on the raw data to compute the distance between cells. You can still do it, but their dimensionality reduction help you to facilitate or accelerate the downstream analysis. So this is after you reduce dimension, for example, you reduce further from 50 dimension to 2 or 3. In that case, we can start to visualize the positions of the cell. So once the cells are close to each other, we can group them based on their distribution on this 2D, 3D space. You can do clustering on 50 dimension space. So after you reduce the dimension, you don't have to reduce further. You don't even have to see the data. You can do clustering on those 50 PC space defined cells. People like to see this type of data because that's for purpose of publications, for visualization. So we can do this 2D, 3D visualization, and you can use this to do the clustering. So after you finish this, we can look at for each cluster. For example, this is brown, and what cells are expressed highly in this group, what cells are expressed highly in that group. So this case, we want to find so-called marker genes. Marker genes is exclusively highly expressed in any given cluster of cells. So those are the examples. For example, AQP2 and these rows of data highly expressed in the blue cells. And the last row, this genes expressed in purple. Once you have those information identified, you can start synthesize your understanding of the biology system. So if you have these genes co-expressed, highly expressed in this population, and then you can assign a cell type to this group based on your marker genes expression. So that's from the matrix to the cluster and the cell type annotation. So that's a very basic step we are going to do before we can compare using cell type specific method. You have to know, okay, this group is this type of cells. And then after you finish these basic steps for two samples, for example, then you can start to compare. In here, I want to compare non-epithelium cell between this group sample versus another. Okay, but you have to do this first. Okay, let's see the technique to do the dimensionality reduction. And then this is a UMI matrix. Rows are the genes, columns are the cells. This is the concept of dimensionality and high dimensional data. You maybe see this before, and if you have a 2D, 3D, and a 4D higher, and there's no way to visualize, you can only imagine what it is. So our data looks like this. So single cell, each sample contains many cells, and each cell defined by many dimensions. So you have multiple high-dimensional data sets in the cloud, and then you want to reduce the dimension to do visualization, to compare, to do the downstream. So sub-technique is essential here. So this is an example to reduce the dimension of a 3D object to 2D. Okay, you can step on this Coca can, and depends on how you process the data, and you can get the different representation when the 3D reduced to 2D. Okay, they're all the same, but the lower dimensionality representation may look very different if you use different technique. Okay, so that's why we need to know different technique. See what's the outcome, what's the principle behind this, and which one we should use in single cell data analysis. Over the years, people found that the t-SNE method has been widely used and which is suitable for single cell data analysis. t-SNE, the technique, is not developed for single cell data analysis alone. It's developed for some others, okay? So high dimensional data in data science field. But then single cell people, when they have data and then realize t-SNE gave you the better visualization. If you look at this visualization, you found small clusters for different potential subtypes of cells. Okay, so this gave you the foundation for your clustering analysis. And we know if you run PCA on this, you will not be able to separate those cells so clearly. So that's why we want to explain why PCA has been invented so many years before, but nowadays people not use PCA for visualization directly for single cell data, but they borrow data science most recent development to use t-SNE to visualize data, because it's a very different approach. So PCA, this lecture will build up on all the previous lectures we learned. We learned from the vector rotation matrix, that's for PCA. We learned about the probability generative model. So to me, this t-SNE most like a generative model. You generate the position of the cells, depends on your understanding of the distribution of a cell-cell relationship in the high dimensional space. Then you pop up those and try to keep their original relationship. So PCA is built on rotation matrix. And the t-SNE is built on generative model, basically. So what's the fate? Fate is based on the distance matrix. Or multidimensional scaling. So they are very different technique. And I hope you still remember what this is, multidimensional scaling. Remember there the map between different cities. You have a matrix of a distance between one city to another. And then I only gave you that distance matrix. You can regenerate the map between cities to keep the distance matrix valid. So that's the MDS. So fate is going to use the distance between cells to build a huge distance matrix. Then using MDS technique to put the cells in the location in a 2D map or 3D map, okay? So we are going to focus on this first, okay? The first half of the lecture. And if you fully understand this, I would say you already covered 25% all the data science and machine learning, what you need to know, okay? So another half may go to the neural network, okay? Neural network is just a matrix multiplication in a different ways. But this is a linear algebra and eigenvalue, eigendecomposition, and covariance matrix, everything linked. So this covers at least a quarter of the machine learning field, the knowledge you need to know, okay? So we are not going to become an expert on this, but once you have the idea, when next time you talk about PCA, you should use more accurate language because you have a deeper understanding of the problem, okay? I don't know who have used that before. Any one of you use the PCA, principal component analysis, before? No, but you heard about this, right? So what you heard about this, when people describe what is PCA, what they say, their definition. Do anyone know? So I commonly heard, someone say, please explain what a PCA is. Oh, I know, PCA is a dimensionality reduction technique to reduce the dimension of the high dimensional data. I say it's half correct, but in principle it's wrong because PCA will not reduce your dimension at all. It's only rotate your data, give you a new angle, new coordinate system. If you want to keep top 2, top 3, top 50 principal components, use this as a reduced dimension representation, you can, but the PCA itself will not give you the dimension reduction. You have to, the PCA is only to rotate your data cloud. Rotate, okay. Allow you to, using low dimension to see more variability among data points. If you rotate the data, you still keep original dimension. You don't have to reduce it. Your data is still the same, okay? So PCA is not technical to reduce dimension, but it's to try to find rotation matrix. So PCA allow you to find the rotation matrix in order to display largest dispersion or variability. Variation in among data points. I would see the between data points. So you actually measure the distance between one data point to another each other, okay? So PCA allow you to rotate your data and then project your data to the new coordinate system and then measure the distance between data points using the new coordinate system. Let's see the rotation. So let's see, we have one data new, okay? So you know this is a one vector. So that's original coordinate system, right? And then you have its coordinates. So you have x and y. This is just one point. So there's no variation between data points. For instance, if you have another one, see, so this is mu1, and you have another data, say mu2. In this case, we have an example of two data points in the 2D space. So what's the variation between these two? You can compute. So along the x-axis is the y-axis. So the variation, so the v, is you can compute. So this is like x2, x1. So that's a coordinate for these two numbers. So this variation, so remember you have different statistics. You can define the variation. You can use variation. You can use a standard deviation. You have a CV. So many different. But in the core of that is you compute the distance. Basically, you say, okay, this is distance. This is another distance. So the variation from 0, okay? So how far away these two away from 0. So this variation is somehow you can compute as a square plus x2 square. So if you consider this as a distance from origin, and then you have basically it's a d1 squared plus d2 squared. So this gave you some idea how disperse these 2 points away from origin. You probably have another one, another data points, which goes to this way, and another goes to this way. So you can always compute the variation by adding the distance of the projection from this point to x. So basically you add more, about x4 squared. They all distance, right? When this distance is large, your total, this variation, will be large. So when you just compute this distance squared, sum of squared, you get a value of describe the dispersion of your data points on the x-axis. Make sense? You can do the same for the y-axis. You just project your point to y and look at how long the projection is. You project to here, and also you summarize those distance by square. That's a variation. That's a dispersion or variation of data points 1, 2, 3, 4 on x-axis or on y-axis. Make sense? You know how to do this, right? So this is called variation between data points or among data points, maybe more accurate. It's not between, because it's among data points to the origin of the center. So you centralize your data. So you just compute how far away the range, basically, is occupied. Now, if suddenly I gave you a new axis, so I'm not going to use this original, suddenly please use this as your new axis. Now I ask you to compute, if you consider this green line as new x-axis, can you compute the dispersion level or variation of your data points on this green line, if you consider green line as a new coordinate system x-axis? Now, what you can do is to start to compute when you project this on here, then you have a new distance, the projection, then project this on green line again, and project this on green line. You change the coordinate system. Now your original data was x1, y1, but now according to the new x-axis, so this number will change. Now you get a new D1, a new D2, new D3, new D4. So now you change to the new coordinate system, so your variation will D1 plus D2 square, D3 square. Depends on how many data points you have on this. So when you compare this green variation with original black variation, in this given case, if you visually summarize those distance, you will see, okay, this green one gave you the higher variance in total. So this will be greater compared to original. Why? Because this is longer, this is longer, and this is longer. The distance on the y, the coordinates on the green axis, all the distance is longer. The projection is longer than original black ones. Black ones was here. This one was here, very short. This one was relatively short, but once you rotate and project on the green axis, you increase the variation, which will make the data looks more dispersed, more separated from each other, from the origin. Is that true? And everybody got it? So that's our purpose. We try to find this new green line, a new access system, coordinate system to allow the project distance to be the largest in total. So that's the purpose of PCA. PCA try to find this rotation matrix to rotate your coordinate system. Either you can consider to rotate the data points or consider to rotate the XY system. They are the same. In order to achieve the largest dispersion, which calculated by the variance of the data points to the center in total. so you can try different new axes. You can rotate 360 degrees until you found, every time you compute the new variation which projected onto it, until you reach the largest, then you stop. That's a PCA. So those are the name. In between, there's called the UMAP. Okay, UMAP is similar to t-SNE. So we're not going to touch on it. So this is an application to change the coordinate system. Okay, I hope you understand this. So if you consider A as original, the x-axis is a horizontal line here, and then A has its own xy coordinates. Well, if you consider the green vector B as a x-axis, that's a rotation, after rotation, then in order to compute the new coordinate system x for A, you need to project this onto B. So this projection can be computed as A, the magnitude of A, the norm, how long this bar is, times cosine theta. And from this equation, we know you can compute this by vector A times vector B divided by the norm of B. So you can easily to compute the projection of A onto B using how long this, so using the dot product. Especially if B is a unit vector, then this part is 1. So the projection, how long this is just the dot product between A and B. So we are going to use a normalized B vector. We normalize it because we want to have the norm or magnitude of B equals to 1. So we simplify the equation. So A dot product of B is the projection of A onto the length of projection of A on the B. So we can use this projection, the length of projection, as the new coordinate. Okay, so this projection computation using this, and it's used to change the coordinates from origin, and it becomes this using B as a new x-axis. So that's why in matrix multiplication, the dot product between two vectors used a lot when you rotate the matrix. Okay, when you rotate the matrix, data point matrix, using the rotation matrix. Okay, and another concept we need to keep on mind is this eigenvalue, eigenvector system. Okay, so for now, this is just a review and what they are. So A is a squared matrix. Then you have this relationship. Now, soon later we are going to use covariance matrix. So we will use CoV. So instead of A will be replaced. A will be replaced with a covariance matrix. Then we are going to do this eigen decomposition. So covariance matrix times x. X is the eigenvectors. So eigenvalue and eigenvector. later we will do a transformation to make this, to put this side, it becomes covariance and X. This tells you, this eigenvector, that tells you how the total of this basically later. So we will maximize this value and to show, okay, this X vectors will be the rotation matrix. In the eigenvector system, X, this is a eigenvector but in PCA we are going to use eigenvector, this eigenvector set as a rotation matrix. Once it rotates the covariance matrix, you get the largest, depends on which set you use. You get the largest eigenvalues. This eigenvalues is estimation of the largest variation. So they're linked. It's hard to understand, but I'll try later to explain this. Okay, so this is an example of eigenvalue, eigenvector. So we have two eigenvectors, okay, and each have this two eigenvalues, and each have these eigenvectors. Oh, PCA example. So this PCA example will have some random points look, but it's generated by a T-pot location of the points on this shape. So in MATLAB, for example, once you have this, you can rotate whatever you want to look at this cloud of data. You can, at any angle, you can see, I want to just reduce the dimension from 3 to T to 2D. And that's you are doing on the screen right now. This is 2D on your screen. So you basically, from any angle, you can stop and look at this cloud of data. But if you apply PCA, PCA will rotate this to a certain angle. Now, when you stop and you reduce the dimension to 2D, and you see the most dispersed overall, the points, okay? So if you see from this angle, for example, if you see, for example, this angle, then you most likely you'll see a cloud of points. There's no shape. But the PCA will help you to rotate this until you see basically from this angle, right? This angle. And now you reduce dimension and you start to see the shape about this similarly random clouds. And if you make this clear, those random dots was generated based on their position on this T-pot geometric position of the T-pot surface. So you can rotate it, but if you allow PCA to rotate it, it will give you a result like this. Okay, why it gave you this? Because on x-axis, so if this x-axis, this is y. On x-axis, you will contain the largest variation between that direction. So largest variation come from the end here and really long from here to here. So that's middle is 0 here, right? So you try to push your points as dispersed as possible. So that angle gave you the maximized separation of those extreme values and also overall. So, And then your second axis is this. This gave you the dispersion from here to here. Okay? So this is an application. So we explained that. So we are doing this automatically when we take a picture for object. If you have a general purpose, just try to display the most features of this teapot, you will take an angle from here rather than from the top or bottom or from the behind. You don't want to 3D data points to be superimposed onto each other when you see the 2D. You want them to disperse as much as possible. So PCA help you to find the rotation matrix to give you the right angle to see the object, to let the data points on the object to be separatable. Okay? This is like 3 genes example. Then you have samples in a 3D space, and these genes gave you those samples. Then you found first PC, second PC, and then reduce the dimension. These 2 PCs gave you a 2D coordinate system, allow those data to be projected onto the space of the 2D defined. And then you have x-axis and y-axis to display the largest variability on x-axis first, then among the remaining orthogonal direction, you found another angle to show from that perspective. Sounds like very complicated. So we need to know how to do this, how to find the rotation matrix. There are many different ways. So over the years, the principle has been established. We will officially use through the covariance matrix at eigenvalue, eigendecomposition of a covariance matrix to find the rotation matrix in PCA. So most of today, we just use computer language to do the built-in function. Many language has this. If you don't care, and then you can just give the data, the function will find the rotation matrix. It will help you to compute you gave the matrix data points before the rotation. The PCA built-in function normally will help you to find the rotation matrix and help you to rotate your data and give you the coordinates, a new system in the new system. So your description, your numerical description of your sample will change. The number will change. And then you can decide, I only visualize first 2D, and it's good enough most time. So it's 3D, and if you do the visualization purpose. And I want you to consider this as an optimization problem. So optimization, we have not talked about this as in the numerical computation field. People try to find the combinations of parameters to solve a problem to achieve optimized solution, as long as you have a standard. So how good, how bad is called penalty function. So you want to reduce or increase likelihood or reduce the penalty. So the optimization in our case will be trial and error method. You randomly pick one vector to see or project your data in there, whether the variation you increase or decrease. If you increase, you keep that, then try a little bit, and make it increase again until you find the largest variation. So this try and error method is optimization. But of course, you want to find you don't want to randomly guess until you find the largest. It takes time. So you want to make the procedure as easy as possible. So there are ways you do the optimization, many algorithms to help you very quickly find the best angle. So I want you understand this because there are many problems you can solve nowadays using optimization machine. You can consider this a very powerful machine, can help you solve any problem, and unless as long as you know a way to quantify the outcome. So for any problem, as long as you know the outcome is good or bad, basically you can solve it using optimization. Like play chess and you know each move, the values. As long as you know how to compute the outcome is good or bad, then most of this problem can be solved in optimization problem. So in our case, yes, this satisfies this definition, because when we select the rotation matrix, we know we have a particular purpose. We want to maximize the dispersion, so we can compute after given rotation matrix, this rotation matrix will do a good job or bad job. So that's why we can use optimization to solve the problem. But for this, because we found the solution to use eigenvector system and the covariance matrix, this saves us a lot. So this just through dot matrix, basically you get the results. Well, here for optimization problem, you have to guess a lot of parameters. If you have a 3x3 rotation matrix, 3x3, then basically you have 9 free numbers, okay? Of course, it's not every number you need to solve, but with the increase of the dimension, you have a large rotation matrix, a lot of values you need to optimize in order to get the best solution. So for PCA, optimization is not the best solution to do it. Well, this gave you the easiest approach. And also, later we will talk about SVD, and you will realize SVD is PCA. Okay, SVD essentially is a PCA. SVD will help you to find the rotation matrix, and also linked with a distance matrix, a gram matrix between data points. So SVD will decompose your data or your data in data matrix. So SVD will be more direct to decompose your data matrix into a rotation matrix and also a gram matrix. Gram matrix is a distance matrix. And in between there, so essentially the PCA and the SVD are the same. Okay, so this is the built-in function in MATLAB. If you do documentation of PCA, you get this, and you put your data matrix in, and you will get the rotation matrix. Rotation matrix is called a coefficient, okay? It will give you this. Well, you can use a covariance matrix, this rotation matrix, to rotate your x to obtain the rotated new coordinate. If you ask score, so score is after rotation of x, okay? It's called score. So score will give you the after rotated values, and you can use the score to plot 2D and 3D space. So the visualization of optimization. So you try different angles to put your new x-axis. So this is the longest. And then if the purpose is PCA, then you should stop. Let's see if I can stop it. You should stop here. You should stop here. No. So this is, yeah, you should stop here in order to calculate the maximized variation. Okay. A question is, once you have this x-axis has been found, then the y-axis will be automatically defined, because for given x-axis you have this direction, then the y will be automatically self-defined by itself. So if you have a 3D, then you need to find x and y, and the last dimension, the coordinate, the x system will be automatically defined, because once x and y are defined, then the z will be orthogonal to both of them. You only have one solution. So their numeric function allow you to do this, the QR function, for example, to do the decomposition, to find that the last dimension for you. So this is optimization procedure in MATLAB. So if you look at this, there's one function called the minimum function search. Okay, so this is a optimization engine. So there's a function to take your object function and try different parameter combinations here until it reaches the largest values. Okay, but it's search minimum, so only found the solution gave you the minimum values. So that's why your objective function, you need to put a negative. So allow it to search minimum value, but it gave you the largest variability, basically. It gave you the largest variability here. And you will see for this small example, you get the PCA solution and this optimization solution will be the same. Okay. Okay, so this is how the PCA will be officially computed through the standard procedure. So we described around and what PCA is, what's the purpose, and compare with the optimization method. But now this is how the real procedure that's in the built-in function is implemented. So to compute the PC means the rotation matrix. We need to do a covariance matrix and to do the eigenvalue, eigenvector system. So we need to revisit how to compute covariance matrix, okay? So this is a covariance matrix, the covariance between two variables, okay? Consider them as two genes, then you have those different cells. Again, covariance and covariance matrix this matrix is between genes. Okay, they are not the distance of similarity between cells. So we go to the variable space to define the relationship between variables, make a squared matrix, the covariance between genes. This is how the covariance matrix is computed. Basically, you are taking centralized data. So A is centralized because you minus its mean somehow in those steps. Then you transpose your matrix and do a dot product between elements. Then you get a covariance matrix. Very easy to compute. You have data matrix, centralize it, and transpose and dot product by itself. Immediately you get a covariance matrix. So that's the description. You have X, N cells, P genes. Then you start to calculate genes average and remove it, so it is centralized. Remove mean, so this step is centralize your data. Now you start to compute a matrix to get a C, which is covariance matrix, has the shape of P times P. So this squared matrix for genes. So this is a gene, right? Between genes. So this matrix compute with a transpose X times X itself divided by N, how many variables, or the sample size. Then for U, for this, you do eigen decomposition. So eigen decomposition is here. So you change the covariance matrix, becomes I don't know, it's a lambda. Then you have to make this correct in order to reconstruct covariance matrix. Basically you decompose it using this eigen decomposition system, and then this is your rotation matrix you want to get. So that's very easy procedure. As long as you know you can describe this 4 steps, that's your homework, okay? Your homework will be, if I gave you a data matrix X, how can you compute the PC, how the PCA is, this process is. So the outcome of the U, this is rotation matrix, okay? So that's the place people start to ask, why does eigenvector of covariance matrix is the direction which maximizes variation of data? So you probably want to click this and lead you to a very active this blog or forum. People are discussing, the technical people are discussing why that's the case. Why just do this, you get such a complicated goal, and to maximize dispersion from the rotation matrix. Why the covariance matrix is play a role in the middle. And this discussion has been reactive again and again over the years, and the new people learn the technique. You always have this question. But if you don't, you just follow this, you will get your computation easily, okay? But I wish you can go into this discussion and learn some of this, and then I'm going to use a small example to show you the link between the computation of this covariance and also y the linked with a variation for your new projected space. Okay. I think we need a 5 minutes break. So I prepare the whiteboard for you. Okay. Okay, so on the screen here is a small example. People use those green dots and as an example for 2D data. So for this 2D, you have a covariance matrix 2 by 2. Okay, because we only need two numbers to define each point. So this covariance and the variance here. In this example, it's using E2, this vector as a new x axis. E2 is eigen system for this sigma, which is sigma. So they try to explain to use this covariance matrix to rotate points, okay? And then derived the formula to do the decomposition of the sigma. Sigma is the covariance matrix. So that's the explanation. Again, your covariance matrix of the data, then take eigen, and then to find the direction which allow you to maximize. So you found the direction of the new axis. So you rotate your data and then project onto the new positions. Then the dispersion on this axis become largest. Again, this is to explain again why the eigenvector of the covariance always is a way to maximize the variation. There are formulas and then derived until this, but still we have a missing part. We need to know this multiple layers. So this is again into the optimization field, okay? So without that, I think we still cannot solve the problems why you optimize the eigenvectors gave you the largest rotation. But we can use a numerical example. So to show you those links, so why the covariance matrix and eigen decomposition linked with the variation, okay? So I'm going to do this on this board here, and this is the example on the on computer. So we have 3 points, V1, V2, V3. That's in our data. 3 vectors put together. V1, V2. So those points V1, which one is V2? Maybe I labeled them wrong. So which one? So this 3, 3. So this is V3 here. So the values have been centralized, right? 1.1. So So this should be V2, the V3, V1 is here. So you centralize your data. Basically, you move the origin in the middle of these 3 points. Okay? So you can compute a covariance matrix from the centralized data. Okay? You know how to compute the covariance matrix. It's between these 2 variables. So that's 2 by 2. Another way to compute the covariance matrix is using this multiplications of matrix. The data has been centralized. So you transpose it and multiplicate by itself and divide by 3. So there's a number of samples. You have 3 points. So this is your C is your covariance matrix. You can see the values are the same as the built-in function calculated. Okay? So that's the way you compute the covariance matrix and you get a C matrix here. Now, let's select a random vector. Use this random vector as your new x-axis. So this is new x. Right? Just consider this as a new x. That means the data points has been rotated that direction. It was the original x-axis is here. Now use that. So that means all the data points, V1, V2, V3, has been rotated this way. Okay. Just imagine this you selected. Now we need to calculate the projection of each point onto this new axis. So you need to do is to project this onto here and see how long this vector is. Project this onto it. And project this onto it. And to compute how long the projection is. So you have this projection, and then this projection. You want to calculate the total of them. How can you compute? You project your data point by multiplicate with E. So this is a dot product. That's the length of the projection. That's how long this projection each is. Then you squared it and multiply it and sum. So this is as I wrote here. Remember, we have a D1, D2, D3 square. So this is your new variation. Of course, we want to divide by 3 by the sample size. So you get a number 0.4138 after you do this. However, you can also compute in this way. You can compute the dot product between the covariance matrix multiplied by E. So E is this vector, and the dot product again onto E. And you get the same number. So this same number tells you you can use this equation to compute the total variance after you use E as a new x-axis or others to project on U. So where the information comes from? The information comes from the C matrix, comes from the covariance matrix, not from any V, not from any data point itself. So what I highlight in this green box is use the dot product, the slide with a star, and I say, okay, you can do this projection to compute projection. So this is a direct compute projection of your data points onto the E, the vector, and then compute the dispersion, the variation, where everything in this box is geometrically explained using that projection of dot product. Where numerically this number you got is the same as the dot in this yellow box. Okay, so that's why you can use this equation, use covariance matrix in the center to compute the total variance of your data point projected onto the new axis. So we need to find a E gave you the largest so we need to find a E. So this E is I just arbitrarily and found randomly by a combination of two numbers to define this. But you can try many different ones. Every time you compute this, it give you the total variance, same numeric. But I know the eigenvector, eigenvalue system can help you to maximize this. Okay, the first eigenvector linked with the largest eigenvalue will give you the largest value of this. So that's why you can compute an eigenvalue system to get maximized this, to reach to the maximized covariance, the variance. Yeah, so it's a little bit tricky, and I can prove this using the small examples. It's very difficult to explain using the theory or this numeric, this formula, but the same idea, okay? If you look at these equations here, the total variance is the same as what we write here, okay? Okay, so let's move on, and there are some good about this covariance matrix, because this matrix is a, yeah, question. This one here? Mm-hmm. So this E is a vector, right? So this E is this. I just randomly select a vector. You can define anything. Give you as an example. So if by chance you select E E as your new x-axis. So the old axis is here. So the old will be, so this vector is your new, the old axis, your x, y. So you have this vector, which is this. And orthogonal, you have this. So that's your original coordinate system, which before your rotation. Then I decide to make a new one. So this, by chance, I just see a new one. I use 0.2, then 0.95. I just by chance to select this. And then my new coordinate system of y should be 0.959 negative. And so this set is mine. It was old, then we have a new one. It's this set, x, y. So I didn't draw the y-axis. I only show the x-axis, which is this vector E. So this is the vector E, that direction. Also, this has not been normalized, so it's longer. It should be in the unit circle. So I can divide by the norm and then to get a correct version, but that's only give you the direction. So using this new direction, I need to compute my data points, projection on it. So if I have a V1 was here, then its original projection on this system was this long, but now I need to project on my new axis here. So the projection become this long. So that's my D. It was D1 old. So this is new. So that's where I put it here. So make how long this is based on the using E as a new x-axis. So this numerical small example just demonstrate I can compute the total projection length using covariance matrix instead of I really project everything onto this. I think we can go through again Thursday using this example and you play and then maybe put more data to see. Okay? But for now, you just remember the procedure and like this simple ones. Okay? You do covariance matrix and take eigen decomposition to find the new coordinate system. That will be the coefficients you will find from the using PCA built-in function. Also, the covariance matrix is symmetric, and all the numbers are the real numbers. So this mathematically guarantee the eigenvalues will be the real numbers and can be used as a coordinate system. So there are some of this foundation of mathematically guarantee. And for example, the outcome you get will be orthogonal automatically because your covariance is symmetric and the eigenvectors of the symmetric matrix are the orthogonal, so you automatically get a coordinated system. Their x and y axes are the orthogonal to each other, and then if you have a high dimension z, will be also orthogonal to x and y. However, there are limitations for PCA. When the system has no clear direction, for example, you don't know in this case it's a hairball, and then you don't know how to separate them, or you have this structure, and PCA won't work very well. You can go along this way or that way, but if you have those like separated, they are not in a shape that allow you to find that. Also internally, if you have more intricate structure like this, there's a sine function. It will make the PCA difficult because PCA is not designed for solve this linear, nonlinear structures of data. As I said, it's only rotate your angle of observation. There's no other transformation on it. So if your data is somehow in high dimensional space, very complicated, then PCA will only look around, and then no angle can be found to give you the best separation of data because it's entangled somehow. That's why if you compare PCA on your left and right is a t-SNE on the same set of data, looks like a t-SNE will be amplified those internal structure of data better. Yeah, so will PCA only allow you to separate the sum of outliers away from the center core of this, but not a structure, no structure you can observe from there. So we are going to explain how t-SNE was used to achieve this. It's pretty much like this, the map, okay? If the real relationship between the stations before transformation, they are crowded. So in the downtown, you may have underground station which very close to each other. We can achieve by walking distance, but in the suburban or neighborhood, and the distance between the stops, the stations become larger and larger. So in order to visualize this more crowded center points, you need some kind of transformation. Just like this, you transform the data and artificially introduce some space in between, allow the detailed relationship between points to be visualized, okay? I don't know if you know Jeffrey Hinton. Anyone knows him? He's a last year Nobel Prize laureate in chemistry. So he's a machine learning people. I think his contribution to neural network allowed him to get a Nobel Prize last year. And his early work also include this stochastic neighbor embedding. So this is the foundation for nowadays the t-SNE. So if you look at this, published in 2003, it defines a probability of being neighbors of two data points in the high dimensional space. It's very easy to compute. It's the distance between 2 points, okay? I and j. If these 2 points are close to each other, close to 0, and it will translate a probability using normal distribution PDF to see, okay, if their 2 neighbors are close to each other, then they are likely to be a neighbor that the probability is like this high. If these two neighbors are far away from each other, then the probability being a neighbor will be low. Okay, so that's the distance. If distance is 0 from I to j, and you have a high probability to be considered as a neighbor. Well, if you are away from the distance is large, d1, so d0, then the probability of these two ij to be neighbor is lower. Okay, so basically it's a numerical transformation to transform the distance you measure into a PDF probability. So this is a Gaussian distribution. This is a PDF. So basically that's all. Okay, and then it also takes a relative. So your two looks like to be a friend in this probability, but it's also divided by I to all the non-j neighbors in total. So the relative, if you relatively more likely, you're more likely to be. Okay, so basically it's a probability. So the goal for this embedding is in a high dimensional space. Your data points, for example, cat with kitten and dog, hovercraft. So if their relationship is close to each other, so your p between the ij is a small relatively, and then you want to transform this relationship to a low dimensional space, you keep the probability being neighbors are the same. Okay, so this is not a direct distance projection. It's a relationship between neighbors probability projection. You want to project those relationships. It was a P and it become a Q in the low dimensional space, but you want to keep P and Q distribution as much as possible similar. So you want to minimize the KL divergence between these PQ values, distribution values. Okay. So when you reduce the dimension to 2D, you still want to keep kitten and the cat to be neighbors, more likely to be neighbors. Okay. So this works for no matter how high the dimension is, as long as you can define the relationship between your points or relative locations compared to other points. If these 2 points to cell, for example, in the high dimensional space, we don't care how high they are, how many genes you define these 2 cells. Every relatively is likely to be neighbor into each other. Then I want to reflect this relationship in the 2D. So I will allow randomly generate this position of these 2 cells to keep them close to each other compared to close relationship with others, because these 2 are very close in the high dimensional. On the other hand, if the high dimensional space, these 2 are far away, and I want to keep that relationship when I project them into 2D. So this projection or embedding procedure is stochastic. We don't keep anything. Anything is generated by random numbers, basically. So that's why the process is stochastic. I can only guarantee your relative position and the relationship will be kept as much as I can based on the distribution, but not guarantee you will be in the same location. So if I run t-SNE again using different random number seed, I will generate very different picture, but the relationship should keep. So the cat or kitten may not be in that corner of the 2D, maybe in the bottom of this picture, but this relationship with others should keep the same. Okay, so when you do this, in this equation, the P and the Q values are all generated using normal distribution. So the distance and this probability and was measured using normal distribution. When you regenerate the relationship in low dimensional, you still use this distribution to see, okay, if I have this relationship likelihood to be this high, I will generate the distance into 2D space in this. So same thing. But then they found, okay, if I use a T distribution instead of a normal distribution, when I regenerate data, the data looks more dispersed from each other. The reason is the shape difference. As I remember I had a picture which the T distribution, so this is a normal distribution, and the T distribution will have a fatter tail, right? Remember this is T distribution. So when I learn the relationship between 2 points, I use distance between these 2 points in the high dimensional space and define their relationship, gave me the relative possible to be neighbors like this. But when I redefine the distance, I project this onto here, This is the same probability. But I generate the data, I use this longer distance. Dij is longer in T distribution. So the same probability, but when I regenerate data, this gave me longer distance between these points, 2 points, because I follow T distribution. So this longer distance gave you more dispersed, regenerated data, okay? It was relatively crowded in higher dimensional space. But when I project on the 2D, I regenerate the position and put them slightly away from each other. The only slight difference is this region, okay? So why we need a T distribution? Because the t-SNE, because if you have this nonlinear relationship among data points, if you simply project this, use the PCA, a lot of those distance will not be directly projected distance will not allow you to separate the points, okay? In this case, if you have the shape of this, you will project it into low dimension space. They all crowded onto each other, but we try to move them around to make them more separable. So in this case, this 2D not project onto X or Y axis, but it's regenerated and put into low dimensional space. There's another illustration and the distance to similarity. So this position, this gray point is close to blue ones. Okay, if you take this as a center, then those ones are close to gray ones by this likelihood, this chance. Well, it's very unlikely to be neighbors from orange and the red points. So this translates to low probability. So that's the relationship between in high D. Then you do a transformation and put the distance the larger. So it depends on the shape, okay? The relative score will be the same. Then that's the difference I just draw on the border. So T distribution, let's give you the fighter tail. So the same probability will translate with longer distance. So you keep the relationship among data points in the low D space. So this is it is an optimization procedure, and to allow you to find the relative position to keep the same relative position of data points between high and 2D, and just when you project them, the distance computed is slightly different using different distribution. Okay, so this is a Tisney, and we are moving to the last dimensionality reduction technique. It's called manifold learning and using the FATE. So the idea is all the cells have a relationship, and you can build a cell-to-cell network. So something looks like this. If you compute the distance and put the cells in a 2D space. So this is a complex shape, and the cells not follow a simple linear layout. Okay, in this shape you can see, okay, there's a structure, and if you build a network and if you trim the network and only keep the so-called KNN, you can transform this. So if it's too long, then you can trim off the link between them. At the end, you get a shape of like this. Okay, you want to learn this shape by using the small steps. You can only move small steps, not jump, not move across the structure. Okay, you learn from that to define a distance more realistically. If you don't consider the shape, you can jump, for example, from this cell to this cell very close to each other. But in reality, if you follow the global structure, you cannot jump from this cell to here directly. You have to follow this, I don't know if you can see, until you reach to this cell. So this is a property of the manifold shape to allow you to follow the global structure of your data point formed, but not by this cross-domain jumping. Okay, you can move small steps, and there's some cutoff allow you to do it, but without the long distance jump. Or you follow that, you compute a real distance from one cell to another by respect to this manifold structure. So that's the idea about this dimensionality reduction, to treat the cloud of data points as a complex manifold structure rather than just simply compute the distance. You know, in t-SNE, we just compute the Euclidean distance between cells and the project using distributions. But here, we think you cannot just simply using the Euclidean distance, but as a case I showed here, looks like the distance of these two cells very small, but in reality, you need to move all the way through almost the whole structure in order to getting from this cell states to another. Similar here, and in this, you just cannot jump to here. Okay, you need to follow the structure of a development. So once you have that idea, and this method compare between PCA, so if you have a structure data, if your PCA gave you this, t-SNE gave you this, FATE gave you almost the structure you got from original. And you have a data from single cell, which differentiation of cells, and there's time series data, and then in different states of cells. PCA gave you this separation of different time points, and t-SNE just gave you isolated the structure of the clusters. Well, the FATE will keep the original developed trajectory. So you see from the stem cell to everything else, it's very clear because you know you follow the development time. So how the FATE algorithm is designed, so implemented. First, we compute the pairwise distance. So this is the Euclidean distance, and transform this distance using this affinity, which is a kernel method. Then learn the global relationship using this random walk or diffusion process. Again, this is another transformation. Transform the affinity is using this transformation to cut off some long distance, and long distance will be basically will be reset and cut, and then using diffusion process to learn from one position to another how likely to be move. So you get at the end you get a distance matrix. Okay. It started with a distance matrix, but learn the process using this transformation, using diffusion matrix process. Then you get the distance matrix, and then use MDS. So as I said, FATE is an algorithm based on MDS. It starts with a distance matrix between cells. The only difference from the direct using distance to do MDS is it goes through this transformation and another transformation procedure to change the distance matrix from Euclidean distance to some of this distance defined with respect to the manifold of the structure of data. Okay. I think that's all for this week's theory, and then Let me know if you have any questions, and we will practice a lot of the things Thursday.